Welcome to microbiology. We're studying an introduction to microbiology course, and last chapter we learned a little bit about how microorganisms grow and what kinds of things they need to grow, kind of nutrients and so forth, and how we may grow them in the lab and maybe even distinguish between certain species and strains by what we provide for them by way of media. Now we're going to talk about controlling microbial growth, particularly in settings where we don't want to have microbes in the clinic and so forth. And so we'll be taking a look at a bunch of different um, issues around that, including even food production and so forth. There's some micrograph of some norovirus, if you've heard of those, causing problems on cruise ships, causing little mini epidemics of diarrheal diseases. Well, I'll start this chapter with a bunch of vocabulary terms that you should be able to define briefly. Um, Asepsis means the absence of significant bacterial or microbial contamination. And sepsis, being its antonym, means there is microbial contamination. So in the strictest sense, asepsis means not contaminated, sepsis means contaminated. But later we'll see that the word sepsis is used in a clinical setting a little bit differently. And so we'll be talking about that. So if asepsis means free of contamination, aseptic techniques means doing things in such a way that we are preserving that non-contaminated, that non-bacterial, non-microbial kind of conditions. Okay, how about this list of vocabulary? Uh, just take a few minutes and see which one of these terms you are not so sure about, and maybe make a list and practice them a little bit. Sterilization means removing all microbial life, and that's an important term. There are sometimes situations where we want to have absolutely sterile conditions, such as in canning procedures and so forth. Commercial sterilization, oftentimes it isn't practical to make sure we kill everything, but we may want to kill, for example, endospores. When we are doing canning of food, we want to make sure that there's no endospores that could germinate and allow bacteria that produce some very terrible toxins to grow inside that container. Disinfection means to remove disease-causing microbes. So we'll talk a lot about in this chapter towards the end about some different chemicals that are used to disinfect surfaces and situations so that we don't have any disease-causing microbes. Antisepsis refers to removing pathogens from living tissue. So an antiseptic is something you might put on an injury to kill the bacteria that might have been introduced into the body. De-germing is a little more qualitative term, removing microbes from a limited area, say, for example, the hands. You may give a quick hand wash, de-germ those surfaces. Sanitization, if you go to buy a meal at a diner, the utensils are said to be sanitized, washed in a hot water dishwasher so that there's minimal microbial presence on the surfaces, but not technically sterile. Biocides or germicides or bactericides, those are chemicals that can kill microorganisms. Bacteriostasis, if a chemical is bacteriostatic, that means it will inhibit growth, but it doesn't actually kill the microorganisms, so they may be prevented from growing for a period of time, but not killed, and they may later grow if hospitable conditions are restored. Pasteurization we've already encountered. We'll mention it again. Louis Pasteur developed this method for removing spoilage microorganisms from foods using heat in order to be able to give a longer shelf life. So when you go to the store and look in the cooler and you want to buy some half and half to put in your coffee, you'll see that it's been pasteurized. That means that it's been exposed as it flowed through a pipe to a certain amount of heat for a certain period of time to reduce the amount of spoilage bacteria so you can keep it for much, much longer. If you keep a gallon of milk in the fridge for a couple of weeks, it'll go bad. If you keep pasteurized milk product in the fridge for a couple of weeks, it will be preserved because the bacteria have been reduced by this heat application. Under set conditions, microbes die at a set rate, and it produces an interesting logarithmic change in the number of surviving microbes. So if you look on the right side of this graph, this axis, that's the actual number of surviving microbes, and we start out with 1,000,000. Over a period of time, it's going to drop precipitously because in this particular case, we're talking about conditions that will kill 90% of the microbes in 1 minute. So in the first minute, 90% are killed. The second minute, 90% of the remaining ones are killed. In the third minute, 90% of those are killed. And so you can see how we're killing off very quickly a lot of the microbes. If you plot it as a log scale, then against this log scale on the left y-axis, we produce this linear relationship which we've seen before when we're talking about log rate of growth, log death in a culture of bacteria. In this case, we're doing something, maybe adding some heat or something to kill some of these microorganisms 90% per minute in this case. The effectiveness of an antimicrobial treatment depends on a number of things. It depends on how many microbes you have to start with. If we have a very large number of microorganisms, it's going to take a lot more minutes of time to kill them than if you have a smaller number. Here we started out with only 1,000 microbes, and in 3 minutes' time we may have killed off 99% or more of those bacteria, whereas we start with 1,000,000. In 3 minutes we still have a significant number of bugs left. Another thing, organic matter. In the clinical setting, especially important, it's relatively easy to kill microorganisms on a clean, bare surface, but if there's some organic material like sputum or vomit or feces, it's going to be much harder to kill those microorganisms. They're sort of protected by that organic material. Temperature, biofilms. If we heat up the temperature, we're going to have a much higher rate of death, a rate of killing of microbes. Biofilms make it, again, much harder to kill. If you take a catheter or an endoscope that's normally inserted into the digestive tract for analysis, and it's got some biofilm on there, you might think you've treated it in a way to make it ready for the next patient, but if there's a biofilm, it may still have live bacteria on it. Time of exposure. The longer you heat, you can see on this chart, the better you do at killing the microbes. Different microbes are easier or less easy to kill, and at the very end of this chapter is a little table that sort of lists hierarchically microorganisms in their order of susceptibility. We'll see how some are more durable than others. Basic principles of controlling microbes, either live or growth. Alteration of the cell wall and cell membrane. We've already mentioned that if you disrupt the cell wall, most microorganisms don't have a method for controlling osmotic pressure. So bacteria, typically their cell wall allows them to have a very high osmotic pressure inside. There's a lot of solutes inside. We don't worry about that because the cell wall can hold that pressure. But if the cell wall is compromised, the bacteria will lyse or burst. Something that damaged the cytoplasmic membrane will kill by preventing the bacteria or microbe from controlling its chemical composition in the cytoplasm. Some viruses have a lipid bilayer, phospholipid bilayer on the outside that's applied or occurs on the surface as they bud out from the host cell in which they were produced. And that lipid envelope, it's called, contains the spike proteins that are needed to bind to receptors on the host cell in order to be taken up by the host cell. So if we can disrupt that viral envelope, then the virus is neutralized. An envelope virus needs that envelope to be able to attach to and enter cells to be replicated. Damage to protein and nucleic acids, lots of different agents that we'll talk about later in the chapter. Chemicals that can oxidize, for example, and damage protein and DNA. Radiation, electromagnetic radiation that can, or gamma radiation that can strike and injure the chemically modified proteins, nucleic acids can kill microbes. Heat can denature proteins and so forth. So we'll talk about a number of these different ways. So physical methods of microbial control. Heat is our first example, and a couple of terminologies here that are used especially in industrial settings. The thermal death point is the lowest temperature at which all the cells in a particular culture are killed in 10 minutes. So if you're trying to devise a process for preparing some industrial substance, maybe it's food or a medicine, the thermal death point would let you know um, what minimum temperature you would need to get rid of the microbes. Thermal death time is the time it takes at a certain temperature to kill all the microbes. So we choose a set temperature that's practical to produce in the industrial setting. How long is it going to take to get rid of the bugs? Oh, there we go. Minutes to kill 90% of a population of microbes at a given temperature. That's our, we said thermal death time. Here we call the slight difference in decimal reduction time, specifically how long it will take to get rid of 90%. You can see that in 1 minute we get rid of 90%, go from 900,000 to 90,000. Another minute, 90% of those down to 9,000 and so forth. And we can keep on reducing the number of live remaining bacteria very effectively using this decimal reduction time at a given heat. If the time is much longer, that might, at a particular temperature, it might take much longer than 1 minute to kill 90%. That would be the decimal reduction time. So moist heat specifically, steam autoclaves are instruments designed to introduce water at a super high temperature under pressure to kill microorganisms. And moist heat is much more effective than dry heat at killing microbes. If you want to sterilize something, you can place it in an autoclave. It raises the pressure, therefore raises the boiling point of water. And standard rule of thumb is 15 pounds per square inch of steam, that's water vapor, superheated water vapor, at 121 degrees Celsius for 15 minutes will sterilize a solution or sterilize an instrument that you place under those conditions. Autoclaving, that's the predominant way in microbiology laboratories to sterilize media, for example, or instruments for handling microbes. Pasteurization, as we've said, reduces spoilage organisms and pathogens. So what you can do is pass a liquid, like as we said, half and half, or a juice or something like that, or milk, through a tube that is heated as it passes through, and we may be able to heat the milk or fluid as it's passing through there at 60 degrees Celsius for 30 minutes, pass it through a series of loops of tubing for 30 minutes as it passes through, would pasteurize the fluid. If you raise the temperature, it'll take less time. If you raise the temperature higher, 72 degrees Celsius, we'll only need to expose the fluid to that temperature for 15 seconds in order to get rid of spoilage organisms. Ultra high temperature, 140 degrees Celsius, for only 4 seconds. So you can see this inverse relationship between the temperature and the time it takes to kill the microbes, but you have to consider what effect the temperature will have on the substance being pasteurized. So some substances would be completely ruined if they were exposed to 140 degrees Celsius. So you would use a cooler temperature for a longer period of time to pasteurize that material, whereas if you have something that's quite durable chemically, you can raise the temperature a lot higher. Endospores might still survive, however, so we have to keep in mind not all microbes can be killed easily by this method of heat. Dry heat, as we mentioned, is less effective than autoclave. At 121 degrees Celsius, you can sterilize a solution, for example, in 15 minutes. Even up to 170 degrees of dry air, 2 hours would be required to sterilize something under those conditions. So moist heat is much more effective than dry heat for sterilization. Some physical methods, other physical methods, filtration. A filter membrane that has pores as smaller than 0.2 micrometers, 0.2 millionths of a meter, would be sufficient to remove most microorganisms. So that's considered a standard method for sterilizing a fluid would be to pass it through a 0.2 micron filter. You can chill or freeze microorganisms in order to prevent their growth. Um, we talked about intentionally preserving specimens of microbes. We can freeze them at minus 80 degrees, or we can lyophilize, freeze dry them, and store them for long periods of time. They won't grow, and then we can choose to grow them as we desire. High pressure can denature proteins, not a very common way. Desiccation, dry things out, it prevents metabolism. Drying is a good way to preserve or inhibit growth, excuse me. Osmotic pressure produces plasmolysis, the loss of water by osmosis, shrinking of the cell membrane down. We've talked about that in the past already too. So if you put microbes in a very high osmotic pressure solution, say with sugar or salt, plasmolysis will occur and no growth will take place. Back to methods of actually sterilizing. Radiation. Ionizing radiation, such as X-rays, very short wavelength electromagnetic radiation, gamma rays coming from nuclear instability and radioactive emissions, electron beams. We talked about electron beams when we talked about the electron microscope, very short wavelength electromagnetic radiation or particles can sterilize. They destroy DNA and proteins. Non-ionizing radiation, ultraviolet light, electromagnetic wavelengths that are much longer than, say, X-rays, can still kill microbes. And here we see this spectrum of different wavelengths of electromagnetic radiation, long wavelength on the right, shorter wavelength on the left. As you go into shorter and shorter wavelengths, the energy that the photons have is greater and greater. And so we see ultraviolet light right here is sort of a cutoff. 280, anything shorter wavelength than 280 nanometers, ultraviolet light range B and C can destroy microorganisms. So down here in this short wavelength range of ultraviolet light, we can sterilize. In fact, our tissue culture hoods in the laboratory use a ultraviolet lamp to sterilize the interior of the hood when we're not using it. Using chemicals, disinfectants are chemicals used to control microbial growth. The concentration of the disinfectant has a big impact on how well it's going to do at disinfecting. The presence of organic matter, we already said, a particular concentration of disinfectant will have very different effects depending if there's organic material mixed with microorganisms or not. pH and time, we'll look at those issues as we go through a couple of different scenarios. How do you test to find out if a particular disinfectant at a particular temperature is going to sufficiently remove microorganisms in a setting in which you wish to operate, say bringing patients in and operating with them? So one possible way to establish the effectiveness of a particular cleanup would be a use dilution test. So metal rings or rods are dipped in a solution of bacteria and allowed to dry, sort of like when you produce a slide for staining. Then we place those cultures, those rods or rings in a particular disinfectant we want to test for 10 minutes at 20 degrees Celsius, and then transfer those rings or rods into some fresh culture media, rich culture media like nutrient broth, and see whether it grows. And if anything grows, obviously we do not kill the microorganisms. Another way of evaluating disinfectants for their effectiveness is to place some disinfectant in a porous paper disc or some kind of spongy disc and place it on a agar plate of media and that's been inoculated with a large amount of microbes and see where the microbes are inhibited from growing. So these clear areas are areas where this microbes, the white area is a lawn, complete confluent growth of bacteria covering the surface, and where there's no growth, that shows that the whatever disinfectant or chemical was inhibiting the growth of bacteria. Chlorine, pretty powerful at inhibiting. Phenol is a pretty strong inhibitor of microbe growth. Hexachlorophene is a disinfectant used by surgeons to scrub their hands before they put on their surgical gloves to try to minimize the presence of microbes. Um, so Staphylococcus aureus can be easily handled by any of these disinfectants. Escherichia coli, E. coli are a little more tough. Hexachlorophene didn't do much good with those guys. Gram-negative bacteria. Gram-positive bacteria are easier to deal with than gram-negative bacteria. Quaternary ammonium compounds are detergents with amine groups, quaternary substitute amine groups, and so that's something that can be used to help prevent gram-negative bacterial growth. And chlorine, Pseudomonas aeruginosa is like a bad player for sure in a clinical setting. They're very, very metabolically diverse microbes that can survive and even utilize almost anything to grow on. Here we see some chemicals that are normally used as disinfectants are not only not harmful, they can actually provide nutrition for these terrible bugs, Pseudomonas. Chlorine, you see, is a very powerful disinfectant, and it is one of the only things that will kill these guys. Phenol is just a benzene ring, aromatic ring with an alcohol attached onto it, and it makes it quite an acidic alcohol, which is good at killing. Modified phenols, O-phenylphenol is even better. Hexachlorophene, we said, it's a polychlorinated biphenyl or bisphenyl compound, pretty good at disinfecting. So again, it's used as a hand washing agent. Triclosan is another cleaning agent used to get rid of microorganisms by disrupting the plasma membranes. So all of these substances are good at. Biguanides are another type of disinfectant that's often used to try to limit the amount of microbes present in a certain setting. We want to keep clean for the purposes of bringing in patients or preserving fluids. Halogens, iodine and chlorine are very, very good disinfectants in the right chemical conditions. So these guys are oxidizing agents, and so they chemically modify proteins and nucleic acids and damage them and prevent microbial growth, kill microbes. Bleach has a compound that can allow this chlorine to be active. Hypochlorous acid or sodium hypochlorite, pretty good at disinfecting, one of the best at disinfecting. Can actually sterilize under the right concentrations, can actually kill even endospores. An iodophore is an organic compound that releases, say, chlorine or iodine over time so that you have a long-lasting disinfectant capabilities. Just so you know what that's talking about. Alcohols. I'm sure you've heard of 70% alcohol or 90% alcohol. You can disinfect quite well with alcohols. They dissolve the plasma membrane, the phospholipids, and destroy the microbes. Here you can see different concentrations of ethanol and mixtures of ethanol with water and how well they do at disinfecting in a period of time. So down here at 50% ethanol and water, 10 minutes, nope. 20 minutes, no, have not disinfected. In 30 minutes, we can successfully disinfect a solution, say, or a surface of bacteria. If we keep increasing the concentration of alcohol, we get more and more capability of disinfection. And so you can see from 70% up to 95% ethanol, we can successfully disinfect it in as short as 10 minutes. Interestingly, 100% ethanol doesn't really work. You need to have some water mixed with it in order to get a good disinfection capability. 70% ethanol is probably the most commonly used disinfectant for just cleaning up around a laboratory setting. It's because it's just as effective as 95% ethanol, at disinfection, and it's a lot cheaper if you're only using 70% ethanol, and water's very cheap to get a hold of. So thinking of alcohols, ethanol and isopropyl alcohol are often used if you're about to get a shot or a vaccine. You'll see that they'll often clean your skin with an alcohol pad and then let that evaporate and then um, inject at that site, and it'll pretty well disinfect your skin surface. I just circled that 70% ethanol there to remind you that's a good trade-off between effective disinfection and not wasting. Heavy metals can prevent microbial growth. You can see this is a silver dime. The mercury dime was a piece of US currency back when they used to use pure silver to make coins out of. They stopped doing that because they got cheap. But you can see how it inhibited growth around the perimeter around the silver. Silver has had popularity as a way of sort of not only disinfecting, but also sort of supposedly being more healthy. And there's people that will consume certain amounts of colloidal silver in order to theoretically cleanse their body of microbes, but there's no microbes in your body where they shouldn't be unless you have a trauma or a surgery or something wrong with your immune system. Surfactants, surface acting detergents can be used for, we've already mentioned a number of them. We're just putting them in a categorization. Quaternary ammonium compounds are amines with organic groups attached, and they're pretty good at disinfection. Various acidic anionic detergents like sodium lauryl sulfate are pretty good at sanitizing, getting rid of the majority of microbes, degerming the surface. Soap is just um, fatty acids can whisk away microbes oftentimes from the surface because it has the ability to separate the bugs from the surface and rinse them away. So quaternary ammonium compounds are pretty good at cleaning up, at denaturing proteins and preventing microbes from surviving and growing. Chemical food preservatives. How do they work? Sorbic acid, benzoic acid, you'll see them on the labels of a whole bunch of foods, calcium propionate. Um, those are just agents that can prevent growth of microbes. So oftentimes we want to preserve food so it has a longer shelf life before it spoils. And we can use those kind of agents to prevent mold and bacterial growth. Cosmetics even, you want cosmetics to not be, you want to slather cosmetics on your skin and be inoculating yourself with tons of microbes. So cosmetics typically have some kind of preservative in there to prevent microbes from growing. Antibiotics. Here's a couple of antibiotics that prevent spoilage in cheese. Antibiotics are antimicrobial substances that are isolated from naturally occurring sources, other microbes. Aldehydes. You've heard of formaldehyde. If you've dissected anything in the laboratory in school, you've probably been familiar with the relatively unpleasant smell of formaldehyde. Formaldehyde and glutaraldehyde are aldehydes that will destroy proteins in microbes. They cross-link the proteins and ruin the functionality of all the enzymes in different protein substances in bacteria and fungi. Gaseous sterilants. This is something worth knowing about. Ethylene oxide is often probably the most commonly used substance to sterilize catheters and plasticware and various disposable, especially instruments, but all instruments for medical use. Even gowns and cloths, personal protective equipment can be sterilized by ethylene oxide in large quantities in an industrial setting to prepare sterile materials or equipment for using in treating patients. Peroxygens, hydrogen peroxide is a pretty good disinfectant agent, and it's an oxidizing agent which will chemically change proteins and nucleic acids and damage them and prevent microbial growth. So that's just a quick introduction to some different ways, physical ways, chemical ways to control microbes in an environment. And for any particular environment, you have to answer the question, what are we trying to accomplish? We need to absolutely remove all microorganisms, or is it practical just to remove most disease-causing microorganisms? So you have to have some goals in mind, and then you can establish a method to systematically control the microbes to the degree that you need to in that setting. If it's a medical surgical suite, for example, we want to sterilize everything and use the most aggressive disinfectants we can. We want to be able to kill endospores in cases like that, but oftentimes all we really need to do is kill the more common pathogens that would be present in a situation like in food service. Um, this is a cool list that shows in the order of susceptibility different types of microorganisms, susceptibility to disinfectants. Again, viruses that have a lipid envelope that need that outer lipid bilayer in order to attach to its host cells are the most easily destroyed. You don't need to destroy the virus particle, you just need to dissolve away that lipid envelope. Alcohols can very quickly dispatch, or detergents dispatch that lipid envelope, and the virus is nullified, inactivated. Gram-positive bacteria are relatively easy to kill relative to gram-negative bacteria. Um, Viruses without envelopes are a little more tough. They have a protein capsule on the outside that has to be penetrated and damaged and destroyed in order to prevent them from being able to get into their host cells. Fungi with their cell walls or fungal spores are pretty tough to kill. Protozoa are rough beasts, single cell organisms, but protozoa oftentimes produce cysts that can persist. That's sort of like their version of an endospore, something that can survive very hostile environment until a nutrient source can be restored and they can re-germinate and grow again. So very hard to kill some of these protozoa cysts. Mycobacteria, those are those species of bacteria that have the mycolic acid cell walls, are very hard to kill. That waxy mycolic acid protects them. Endospores of Clostridium and Bacillus bacteria, very, very hard to kill indeed. So that's the gold standard. If you have something that can kill endospores, a system or a method that kills endospores, you've really gone the extra mile to sterilize the conditions. Prions or prions, we won't really talk about those right now, but we will come to those at a later time. They're just individual proteins that can be infectious and cause disease, amazingly. They're notoriously difficult to destroy. So if you do have a situation where you know that there's potentially exposure to bovine spongiform encephalitis, for example, prions, then you need to really have some harsh chemicals to make sure that those proteins couldn't survive. And, here's a list of some different methods or disinfectants and how well they do at killing endospores. So that's the toughest thing that oftentimes we need to deal with. Mycobacteria often pretty tough too. And so you can see that, let's look for chlorine and iodine. Chlorines, probably the best disinfectants, can kill endospores, but you've got to make sure you have a sufficient concentration and duration. So at best, they're only fair. They're not as good at autoclaving, for example, as at killing endospores. A lot of very practical disinfectants that we use all the time are not able to kill endospores. So quaternary ammonium compounds and bisphenols that are often used for washing up before surgery, for example, they're not about killing endospores. So you have to keep in mind we're not sterilizing the skin. We're just degerming the skin, and then we'll put on some sterile surgical gloves to make sure that the last remaining entities can't get into the wound. All right, that's it for chapter 7, and I'll see you back here next time as we move on in our text to chapter 8.